DNA replication. Hello and welcome everyone. Today we are going to talk about DNA replication in prokaryotes. Prokaryotes have circular genetic material, that is the DNA or RNA, and this DNA replication in prokaryotes are organized in 3 steps that are initiation, elongation and termination of the DNA replication. It has only one original replication where the pre-replication protein complex will bind and cut it, forming a replication bubble. Next, the helicase enzyme will bind and unwinds the helix, and single-stranded binding protein will bind to the DNA strands, preventing them from base pairing again. For the supercoiling, topoisomerase 1, having ligase and nuclease domain, will cut the supercoiling and reseals it. Next step, the elongation of DNA. RNA polymerase, the primase, will form a primer which will provide the treatise and to the DNA polymerase III. DNA polymerase III responsible for the normal DNA synthesis in bacteria and adds nucleotides for the synthesis of the DNA. Since DNA is only be synthesized in the 5' to 3' direction, only one strand can be synthesized continuously. This is the leading strand, and the other strand is discontinuous, called the lagging strand, and synthesized as short pieces, approx 1000 bases long, called the Okazaki fragments. DNA polymerase also has exonuclease activity, and it checks for any error and replaces the mismatched nucleotide, replacing it with the correct one. This is the proofreading property of DNA polymerase. Next, DNA polymerase 1 will be binding and removing the RNA primer and replacing it with deoxyribonucleotides, and the final gaps are sealed by DNA ligase. Replication stops when DNA polymerase hits a primer, and topoisomerase 2 cuts the link circles, allowing them to disconnect. So this is it for the video today. Thank you everyone, and if you like this video, please subscribe to our channel, like this video and share among all.